a shaker incubator are flasks holding insect cells, and we can give them the instructions to make a protein we're interested in studying using a Baculovirus Expression Vector System, or VEVS, which makes a baculovirus, a virus that's going to infect the insect cells and deliver the recipe for making the protein. They'll make the protein, and then we can study it. But it's not quite so easy. So it's more complicated than expressing or making a protein in bacterial cells, but it works better if you're trying to study something that's more complex. Maybe it's bigger, it has some modifications, it needs help folding. So if we're trying to study like a bigger human protein, it might not express that well in bacteria cells. And so insect cells could be a great alternative. They're more human-like, at least they're eukaryotic. But they're cheaper than like if trying to do mammalian cells. And how it works is that we give them the instructions to make the protein that we want to study by making a baculovirus, which is a virus that infects insect cells, but not other types of cells. So we don't have, although we take like safety precautions, we don't have to worry about it like infecting us while we're working with it. But how it works is that we need to make this virus, and the way that we get this, we need to make the virus that we can get into cells to make more of the virus and to make the protein. So in order to do this, we have to go through this like back-mid making process, where basically we take the instructions for the protein and we clone them into a like a special type of plasmid. So it's like the plasmids that we use in bacterial expression, except that we can combine it with this like back-mid, which is like a plasmid, but it can grow in bacteria or in insect cells. Then that back-mid has the instructions for making the virus, but it's not the actual virus, so it doesn't have the code and everything that's needed to actually get into cells. So then we have to get the back-mid into the cells using transfection, which is basically where we just like force the DNA in. Then the cells start making the virus, they secrete the virus, we collect that virus, we put it on more cells, those cells get infected, those cells start making our protein, then we can purify it out. So it's like a long process. It can take like a couple of weeks from like starting of your cloning to like actually getting protein to study. So right now I'm in that like waiting period, and it's like, I want my protein. So I thought I would tell you more about the Baculoviral Vector Expression System. So I guess it's actually a Baculovirus Expression Vector System. No matter how much I work with it, I can never seem to say it correctly. But so what do all these words that I can't put in the right order mean? So a baculovirus is a virus that infects insect cells. The name is kind of confusing because it is not a bacteria. The baculo part comes from that it has this like rod-shaped look. And what's cool is that kind of like bacteria and the working with bacteria plasmids and stuff, where they have these circular double-stranded DNA genetic information, so does the baculovirus. And so that makes it easier to work with. Expression, that just refers to like making the protein, basically. Vector is like a vehicle that we can use to get the genetic information into cells. And then system. So a little tech note. So usually these baculoviruses that we work with are based on this Autographa californica multiple nuclear polyhedrosis virus. And I thought that was hard to say. Or Bombyx mori, the silkworm nuclear polyhedrosis virus. And the key word for us is polyhedrosis, because we're going to see later that there's this polyhedron protein that we're going to take advantage of, its promoter. As for the cells that we're infecting, the one that our lab uses the most is Sf9, which comes from this fall armyworm, Spodoptera frugiperta. That says Sf21. And then sometimes we use Hi5 cells. I don't really like Hi5 cells. They're from a cabbage loper, Trichoplusianae. So this works better for some proteins, but it's also like goopier, and I don't like dealing with all that goop. And so anyway, let's go back. So I'm going to give an overview, and then I'll go into more depth into how this is actually working out and how we're kind of hijacking these insect cells and what it looks like in practice. So we start with the subcloning. So subcloning is basically where you're taking something you've cloned. So you've taken the genetic information for making the protein out of its like normal home, and we put it into somewhere else. And so then we stick it into somewhere else. And so here we're sticking it into a donor plasmid.
And so this donor plasmid is basically, it's going to allow us to make a backmid. And so the backmid is going to be like a shuttle vector. And that shuttle is meaning that it can like kind of grow in different types of cells. So we can grow it in bacteria cells and we can grow it in insect cells. And so we're going to kind of like make it and make lots of it in bacteria cells, which are really easy to work with. And then we can stick them into the insect cells once we've got a lot of it. And so how we're going to make this backmid is basically we have this acceptor backmid, and we want to stick our gene or the cDNA, the instructions for making the protein, into this acceptor backmid to make a backmid that has our gene in it. And we're going to do this in a way that's going to allow us to see that we put it in there. So we're going to use, we're going to stick it into this LAC-Z gene. And so the LAC-Z gene is basically going to allow us to do blue-white selection, blue-white screening. So I talked about this the other day and I have a bigger post on it, but basically you stick your gene into the place that's needed to make this beta-galactosidase, part of the beta-galactosidase protein, you interrupt that, and then the bacteria can't make this blue thing. And so when you can't see the blue thing, then that tells you that that colony does have the insert likely. And so then we pick those white colonies and work with them. And so that when you get success, then you see these white colonies and you take them and work with them. And so this is your backmid, or at least there's a bacteria containing your backmid. You have to actually purify it out. And so we do it kind of like a mini prep, except that we don't use columns. So we use like homemade, like all the like P1, N1, N3, whatever, those like proprietary formulas. We actually make them ourselves, and then we just don't use a column. And so we spin things down and we take things off, and we do this in like, do the end parts of it in like sterile hoods. So we keep things sterile except for the virus that we want. And now at this point, we need to get it into insect cells. So we don't have virus at this point. We just have like this information for making the virus. When we have an actual virus, it's going to have like a coat and everything that it needs to actually get into cells. But at this point, it doesn't have that. And so we need to actually like give it help getting into cells. And so here we're going to use transfection. So transfection is just like basically a fancy word of saying we're going to stick genetic information into cells. So if you work with bacteria a lot, you might be used to the term transformation, which is where we're sticking things into bacterial cells. But when we stick things into other cells, genetic information into other cells, we call it transfection. There are a bunch of different transfection methods. And when we're working with this, we're typically using some sort of cationic carrier, so like a lipid, and with like a positively charged lipid, and this is going to help give it into the cells. So we use like there's like transfection reagents that you use, like a mix of chemicals, like these charged lipids and stuff. I'll go more into detail on these various steps later. This is just an overview. And so when you transfect it, basically you're going to stick this backmid into the cells, and this one on the plate, and then these cells are going to start making the virus, and they're going to secrete the virus, which is really cool because this is now it's actually making the real virus with like the coat and everything that it needs to get into cells. So then we're going to collect that virus, but we don't have very much of it, so we want to make a lot more. So we use this virus to infect more cells, this time in flasks, and then those cells start making the virus. So we have a lot of virus now, but we want a lot of protein. And so now we take the virus and we infect a lot of cells, and we let them grow and grow and grow, but not too far that they die. And we let them make our protein, and we monitor their making the protein. We can do this using by watching like a reporter, like a YFP gene I'll tell you about. And then we can see that the protein is getting made, hopefully. And then we collect we stop their growing, we remove all the liquid we call media, and collect the pellet, so all the cells, and then we stick them into the freezer. And then later we can break them open or lyse them and purify our protein. So normally when we are doing this growing, we're doing them in like 1 liter flat or 2 liter flasks. So we just fill to like 1 liter or less, because you want to give them lots of air to grow. And so this is what we normally use. There are times where you need to grow a lot. You need a lot of protein. And so there was a time when I I needed a lot of protein. And so earlier in my PhD, I was using these wave bags.
This is the air pump pumping in the air, which goes through filter. In here is the media and the cells. This is the out filter. It has a warming pad to keep it any vapor from condensing. This is where we put the media in. This is where we're going to take the sample out at the end. This is so that we can take out little samples to test and see how much cells are actually in there.
I was doing like 8 liters there, but sometimes you need a lot of protein. So like in industry, they have like giant vats and stuff. And so one place where this is used is in like viral protein nanoparticle vaccines, like Novavax's COVID-19 vaccine. They use insect cells. They use this system to express the spike protein of the coronavirus and then purify out that protein in the form of like these lipid, these like little particles, so little rosettes. They have this like detergent ring that replaces the membrane. So you can use this system. Typically, when I'm doing it, I'm looking at proteins that are soluble proteins, like cytosolic proteins that are inside the cell. So I break the cells open and keep the insides, but you can also purify proteins out of the actual membrane. So you can use this system to study membrane proteins as well. And I'll get a little more into these various things later. But let's go back and look at how this system is actually working. So let's start with where it came from. So basically, baculoviruses are these natural viruses that infect insects in the wild, and we unnaturalize them to turn them into custom protein-making factories. And so, as I was mentioning before, they have these circular double-stranded DNA genomes, which makes it nice to work with. And so their goal is to get into insect cells and get this insect cells to make more of them. And they can do this in a couple of ways. So they have this like phases of their life. And so one way is that early in the infection, they start making and secreting this budded virus. So this virus buds out of the cells and it has the membrane and it has these surface things that it needs to get into other cells. And so it does, and then these cells start making virus. And these viruses, then they use this to they're jumping into other cells in the same insect at this point. So they're going from cell to cell into the insect, and eventually the insect gets so sick, and then it can't support the virus, which isn't good for the virus. So the virus has to switch strategies. So late in the infection, the virus starts making a sturdier form of the virus, and this form is going to be able to survive even once the insect dies. So it makes the sturdier form, and then when other insects, it lyses the cells open and stuff. And then once the insect dies, and then another insect comes along and it eats like a leaf or whatever that this insect that was there had, the dead one was inside there. So basically you have this really sturdy virus that survives after the insect dies, and now other insects can get infected with it. And so it has to be really sturdy because it has to survive after that other insect died. Now it needs to be able to survive after this this new insect digests it, and then once it's like in the gut and stuff of the insect, then it gets broken open and the virus escapes and can make new virus. So that sturdy shell is made up of a protein called polyhedron. So I told you that polyhedrosis thing would come in handy. And some other stuff in that shell. But basically, we can then take advantage of this. We can take the gene for polyhedron, which they only need late in the infection. So they can bud fine without this. And especially because they need to make a ton of it to make the occlusion bodies. And so those were like those sturdy like things that sometimes called like occlusion bodies. So when they're making those, they basically have to devote like all their time and resources to making it. Which isn't good when they want to do other stuff. So they normally don't make it until they need it. And so they control the expression of this. And so this is going to give us a way to kind of control the expression, but not really, but to get lots of protein made by replacing the gene for polyhedron with the gene for our protein. So early in the infection, they're not making it because they're in this like cell hopping, like hopping from cell to cell in the same insect. But later when they want to do that host swapping, they're going to start making this. And so when they would normally start making this, now they're going to start making our protein. And so we replace the poly-H gene with the gene for our protein. And now when these cells start making our protein, when they think they're making polyhedron, which they only do in this very late phase. And so in this way, our protein is going to be made in this late phase, but it's not like we're making it on demand like we do with bacterial cells. So bacterial cells, we like induce the expression. We put it in, we have this inducible expression system I talk more about in my post on bacterial expression. But you can add IPTG and then the cells start making the protein. That's not the case here. Instead, here we are actually like controlling the like promoter availability and stuff. And so, but in this baculovirus system, we're just using how it normally does things. We're just kind of like hijacking its normal system. And so you can think of it kind of as like a TV channel, like some on-demand programming. Instead, what we're doing here is we're taking what show would normally be on in a certain time slot, and we're just sticking a new show in there. And so now at that time slot, you were expecting it to be Olympics, and now there's the Super Bowl. And so it's that sort of thing. We're just taking a different thing and putting it in the same place. And so we have to go, it only gets made when the other thing would normally get made. We can't just on demand say, okay, make this. So how this is actually getting controlled in both of these cases is by the promoter. And so the promoter is controlling things at the level, we can talk about expression at the various levels. And so ultimately we're talking about like protein expression, but in order for the protein to get made, the genetic information of it, so in this case, our cDNA that we put in, it has to get made into messenger RNA, which has to get translated to make the protein. To make that messenger RNA, it has to go through transcription, and this transcription is can be regulated by the promoter region, which is a region upstream of the region to be copied in to make that messenger RNA. And so there can be transcription factors that can bind to promoter sequences in front of the gene to recruit RNA polymerase. So in the case of bacterial expression, we were kind of like hiding this promoter and then releasing the promoter when we, freeing it up to get transcribed when we want it. But here what we're doing is we're basically just sticking our gene in front of the normal poly-H promoter, so this polyhedron promoter. And so in order to do this, we are going to use these special cells. So the first step is like subcloning. Your gene is going to be in some sort of plasmid usually when you're starting. You've already cloned it into some plasmid, maybe like a DH5 alpha or something that's good for making lots of copies of the gene. But now you need to get it into this special donor plasmid. And so remember, plasma is just like the circular piece of DNA that are for bacteria. We need to get it into a BACMID, which is like this shuttle vector that can be grown in both bacteria, and it can be copied in propagated in bacteria, but then it can also if we put it in insect cells, the insect cells can host it and use it to start making virus. This is the system that we use, a good article on it I will link to, but basically in this system, you have this donor plasmid and then you have an acceptor BACMID. There are various acceptor BACMIDs and donor plasmids and stuff, depending on your wants and desires and how many genes you want to clone. Like you can do like multi-bac where you put multiple genes in. And so here, the one I have is it has this TN7, these transposon sites on either side, and then so does the acceptor, this baculoviral DNA. And so what's going to happen is that when you're doing this cloning, the T7 transposase, so it's actually going to be made by a helper plasmid that is in the bacterial strain you're using. So you need to have the donor plasmid, you need to have the acceptor BACMID, and you need to have the special cells that have the helper plasmid, which has the transposon. transposase. And so because then like each of these plasmids is going to have like their own antibiotic and stuff, and so there's a lot of antibiotic selection going on when you're making this BACMIT to make sure that you have like all the right pieces. But so the plasmid is in like certain cell types. So like we use DH10 multibac, which has this transposase in it that's going to allow us basically to take the sites where they have these matching sequences and to kind of like insert what was here into there. And so we're going to do this so that we stick it into the location of this LAC-Z gene so we can use that blue-white screening. But we're not just going to stick in our gene because you can just stick in your gene, or you can stick in multiple things. And so sometimes you want to stick in multiple, like 2 proteins that you want to study both of them. Maybe like 2 proteins of a complex or 2 proteins that they normally work together. And so one stabilizes the other, that sort of thing. So they might express better if you try to express both of them. Sometimes also you kind of link your proteins together. So they're made in one gene, but you make 2 things at once. But anyway, when I'm doing things, I'm normally just wanting to look at a single protein. But I can take advantage of the ability to stick in the gene for another protein in order to give me a sort of indicator, a reporter, if the cells are making protein. And so I can't really get a reporter of whether they're making my protein unless I were to like tag my protein with a fluorescent protein directly. But if I stick the gene for making a fluorescent protein, so we use this like YFP, if I stick it into a place where it gets expressed at the same time as the one that I would normally get in the virus. So we're looking at like the baculovirus's promoters. We're talking about the polyhedron is needed to make those sturdy like occlusion bodies, those sturdy shells. And so the polyhedron is going to make up those sturdy shells, but that's not all that's needed in order to form these things and then break out of the cells. So you need some help like assembling everything and lysing the cells. And so there's this other gene P10 that's important for that. But because the cell needs to make both of these at the same time, their promoters are regulated similarly. So they both come on at the same place in this like very late phase. So what we can do is we already took the Pol8. So we took the polyhedron and we replaced the polyhedron gene with our gene. But now we can take this P10 promoter and we can take the gene that was in front of it, the P10, which we don't need until the very late phase. And then we can replace it with something like a fluorescent protein, in our case, YFP. And so then when we see this YFP get made, then we know that the virus got into the cells and they've entered the very late phase, which means that our protein is hopefully getting made. And so this allows us to help monitor when it's time to whether the virus is getting into cells, whether they're making the cells, the protein, and when it's time to like collect them. And so you have to take, thankfully we have these really great lab technicians, Sumati and Matthew, who do most of our cell culture work. I did a lot more of my own expression stuff earlier in my PhD. But basically you have to count the cells and monitor the cells, make sure they're healthy, that they're not getting infected. And so you use this this cool little slide, and then you count the cells. You want to make sure that they don't overgrow. And so you monitor like the optical density, or basically you count like how many cells are on one of these little grids, and then you multiply it by these numbers that I can never remember and I always have to write down. But once they get too crowded, then you dilute them. So you like split the cells. So you like pour in more media to double the volume, and then you split them into multiple flasks because you want to keep the flask volume low so that they have lots of room to like aerate and that sort of thing. So it's really important to monitor the health of these cells because and make sure that they don't get infected with things. Sometimes you'll see like the media gets cloudy. One time we had this thing that was like weird. Yeah, we had some weird fungus before. But when we talk later about like cleaning the flask is really important. But it's really important to monitor the health of your cells and make sure that they're not overgrowing and that they're not getting infected with things. Because unlike with bacterial cells, we don't have like antibiotics. We don't have like stuff in here. We're just letting these cells grow, and so we're working a hood with sterile conditions, and take care not to purposely do anything that was going to introduce pathogens, and then we monitor them. So later we're going to be monitoring them for GFP expression. So when we're doing later we'll see once we've transfected and once they start making the cells, we're doing these in these plates. And then we monitor to see whether the cells have gotten infected and whether they're making protein. And then later when we're doing our expression, we'll hopefully see that we get this bright yellow liquid, which is fun. But remember, this is only if you have like a YFP in the thing, or if you sometimes people put a YFP tag onto their protein. But if you don't have YFP, then your cells won't look bright yellow, or and don't worry, that's okay, because that's not going. You don't have the instructions for making a bright yellow thing. Okay, so that's later though, because we need to still get it into cells. And so this type we have a Bachman. And we use cells that are from insects, but we're growing them in dishes and later flasks or bags. And so we start with plated cells because it's harder to get the back mid into the, like when they're floating around freely, and then later we'll scale up. And so we plate these cells into the dish. And then now we need to get the back mid in, and the back mid DNA needs to get the coat to get into cells on its own. So we have to help it out at this point. And so now we add the transfection reagent, which is like that lipid and cationic lipid. So basically cationic, it's got this positive charge that's going to help kind of like neutralize the negative charge of the back mid DNA and help it get close to the cells. And then it's got lipids that are going to help it kind of sneak in between the membranes. And so when we add this transfection reagent, the back mid gets into cells. Some of them will take in the back mid and start making budded virus. And then now it has the coat so it can get into the uninfected cells and infect them. And then those start making and excreting the virus, and they're excreting all of this into the liquid that they're in, so like the media. So the cells are staying on the plate, but the virus is getting secreted into the media above it. But we only have like a couple mils of it at this point. But we only have like a mil or so of it at this point. And so we want to collect that secreted virus to make more virus. So at this point, the cells are still stuck on the plate. So we can just like suck up the virus. We call this the V0. And so we take this little like syringe and we suck up that virus. Oh no, a cell got in, but don't worry. So now what we do is we syringe filter it. The virus is going to come through, and then you don't have to worry about, you don't have the cells in there. So this is the B0, and you keep it in the fridge until you're ready to use it. And so use it for what? Making more virus. So basically, if we want a lot of protein, we're going to need to infect a lot of insect cells. So we're going to need a lot of virus. So our next step is to make more virus. But now we can do it in a flask. So like in suspension, because the virus can get in easier than the back mid could. And so we can take this V0 and we can just add it to some cells in a flask. And so we can't add it to a ton of cells at this point because we don't have enough virus to infect all of those cells, or to at least have enough efficiency. So typically we do this depending on how much we want to express later. We'll do like a 250 mil or 100 mil or up to like a liter for our V1. And so we infect the cells, the virus gets into the cells start making the virus. And now we want to collect that virus, but we need to separate it from the cells. So before, we basically just stuck it through a syringe filter. But since our cells, after we like sucked up the liquid, and then the liquid shouldn't have cells, but in case one like floated off or whatever, then we can syringe filter it. But here our cells aren't stuck down, so we use a centrifuge to help hold them down. So basically, we just use the centrifuge to separate the cells out. When we spin this down, the cells are going to get pulled to the bottom. We pour off the liquid, the supernatant. So now this we have the V1. So this you can keep the pellet if you want, if it's like you do a bigger V1. There'll be some protein in there. Sometimes it's enough for what you need. But if not, then you go to a larger scale expression, which is what I always do. But your virus is in this V1. And so now you can vacuum filter it. So instead, before we just had a little bit, we can kind of just like squeeze it through one of those syringe filters. Now we use this vacuum filter. So we use a vacuum filter when we're purifying, when we're what do you call it, when we want to filter like buffers and that sort of thing. In that case, we use this like reusable filter that we can screw onto different bottles, and we just like hand wash it. And it's okay if there's a little bit of someone else's like KCl or whatever, and you have NCl, like you rinse it off. If there's a tiny little bit, it's okay. But here we want to make sure that we need to keep things really sterile, because remember, we don't have like antibiotics or anything. We want to keep this one virus, but we don't want to keep like, we don't want bacteria or fungi or anything to come in. And so we need to keep things sterile. And so we buy these like single-use filters. So you can actually screw these onto different bottles as well. But they come with their own bottom. So you have this plastic bottom. For the smaller ones. For the bigger ones, we normally just do it onto our own flasks. Okay, so now we have the V1, and now we want to infect a lot more cells so that we can get them to make protein. So before we were after the virus, and now we're after the protein. And so we take that filtered virus, and which we like store in the fridge. And it's really important that you put the dates onto all of these things. So you because over time they'll get not as usable, not as virus effective-y. And so you want to date everything. You can kind of push things a while, but typically just like a couple months at most. And so when you do this, now we have the C1, we stick it into cells. So now we want to do the same. Now this time we want the pellet, because that has the protein. And so we stick into cells, we spin it down, but this time, instead of taking the virus, now we don't care about the virus, now we care about these cells. And so what we're gonna do is we're basically just going to dispose of this virus safely. So we put it in like some bleach and stuff, let it sit, and then we can pour it down the drain. And it's this pellet that we really care about. So how this looks in practice. So my mom took this video a few years ago. And so basically this is me like scrambling frantically, taking all of these like 1 liter volumes from these 2 liter flasks, pouring them into these centrifuge bottles. That which you want to make sure you balance. You don't have to balance them like as perfectly as when you're doing like an ultra centrifuge and like really weighing things out, but the volume, like eye level it to make sure that you're not like doing anything wrong. We don't spin these at as fast as like bacteria cells and that sort of thing. We do it at 4 degrees Celsius. So insect cells are more finicky and fragile and sensitive than bacterial cells. So that's just something to be aware of. So now while they're spinning down, I'm preparing the resuspension buffer. And so basically right now they're in the growth media. And so that growth media is going to have the secreted virus. It's going to have whatever was in the growth media. It's going to have whatever, who knows, whatever the cells were like secreting or whatever. We don't want them to have that, because like part of what's in there and stuff is going to be stuff that the cells were trying to keep out of themselves. And so later what we're going to do is we're going to break open the cells. We're going to lyse them. And we want to make sure that they're coming out into a nice clean environment that's like free from proteases, so free from protein chewers, and that the protein chewers inside the cells aren't going to start chewing up the proteins. So we put like protease inhibitors into the mixture, and so it's basically just like a buffer with some protease inhibitors. Sometimes we put some glycerol in there, depending on the protein and how sensitive it is to that sort of thing, and like a reducing agent, so like DTT or BME or something. And now, once they're spun down, you want to look at the bottom, make sure that the pellet is actually like solid pellety and it's not like coming off the bottom, or it's not fully pelleted yet or whatever, in which case you can spin it down more. But then you pour off the liquid into the bleach. Now you want to keep your pellet on ice. you want to keep everything on ice at this point. And now you put some of that resuspension buffer into the bottle. And now you need to resuspend it. So basically you have that pellet and you want to now like unpellet it and coat it in this fresh liquid. And then we use like this vortex, so you like pipette it up and down. You can use the vortex to really help resuspend it and make sure that basically your goal is to get all of the pellet in there, because you want to get as much protein as possible. And so typically I'll do it so that I have like 25 mils or so per liter. So typically if I do like a 4-liter expression, I'll have 2 tubes. You don't want to fill the tubes all the way full though, because then when you flash freeze them, you are going to, so you put them in, dunk them in liquid nitrogen. If you fill them too full, then they're going to break open, which is not fun, especially if you don't realize there's a crack, and then you go and you stick it in your water bath later to thaw it, and it all starts coming out, and then your water bath is all gross, and then you have to clean the water bath, and yeah, personal experience. So, and now it's time, speaking of cleaning things, now it's time to do some washing. So it's really important that you wash your flasks well, and that you wash them quickly after you do things. They'll get this ring, like this crusted ring, where when the cells were swirling, like the top of where the cells was swirling, they're going to make this like crusted ring of like dead cells. And if you leave that on, then it's going to cause problems when you're trying to use these flasks later, and it makes it more likely that things will get like infected and all of these problems, and you have weird stuff growing. So that's not good. And so you want to wash them right away before anything gets like crusted on. So we have this big brush that we can stick in, and then we wash it. I find it's easier to wash it in like our kitchen, which is we call like our room, with like the center with the autoclave and everything. That's this nice big sinks that are really good for this. It's got like 2 sides. You can do the scrubbing on one side and the rinsing, and put the clean one on the other side. Yeah, I worked out like a whole system, because as you can see in this picture, some days I was doing a lot of washing. And so even this, then after your hand scrubbing, now it goes into like the dishwasher. So we have a dishwasher, but you have to hand scrub them first to get all that cruddy stuff out, and then you can kind of just like wash it. Then it goes to the autoclave, which is like this high pressure, high temperature steamer thingamabob that's going to sanitize everything and then dry in the oven. And so then our amazing media maker, Adriana, she is then sticks the she went before going to autoclave and stuff, she sticks the caps into like these autoclave pouches, and puts foil over the tops of the bottles so that they'll stay sterile. And then when we can go to use them, we just grab a cap and a flask. It's what we do is we actually put the date and stuff on the flasks so that we don't use them too long, because the longer you use them, the more likely it is that they'll get have stuff grow and stuff. Also, when you're taking the caps, they have this like first of all, don't get soap on the caps. They have this like film on them that's gonna like degrade and stuff. And speaking of that film, you want to like hold them up to a light, and you shouldn't see any holes in them. Those holes can then like let things in, and then things can grow. So it's a lot cheaper to use a new cap than it would be, to like throw away an old cap, and even if you have to use a new cap, than it would be to have your whole flask infected or with something that you weren't trying to infect it with, and then have to throw that all away, and then have to deal with potentially contaminating other flasks and that sort of thing. So not good. And so then later though, so once we've frozen the pellet, we can like stick them in the -80 freezer, and then later when we're ready to purify the protein, we can slice them, so break them open. Then we ultracentrifuge them. So before we were just like centrifuging them, now we're ultracentrifuging them, so we're going really fast. When we go really fast, now we can separate like the membrane bits from the soluble stuff. So assuming that you want to study the soluble proteins, you take this protein fraction. If you want to study membrane proteins, now you're going to take the membrane bits. And so in one of my rotations as a lab student, so we like rotate in different labs, and I was rotating in this lab that was studying membrane proteins here for Akawa's lab, and which I had a great time, but I had to switch my mindset from thinking about studying like proteins that were in the membrane versus proteins that were in the liquid. So I was like, wait, I need to keep the pellet this time. Wait, no, I don't want to keep it this time. And so, yeah, it got a little confusing. But membrane proteins can be made using this system, and this is key proof of this is like this Novavax vaccine and stuff, or what they do is they kind of use detergents to extract the protein from the membrane, to kind of replace the membrane with this like detergent, which is kind of membrane like, but now you get these nanoparticles that can be used for a vaccine. So that's really cool. But in my case, I'm just studying the cytosolic proteins, so the soluble stuff. But with regardless of any of your cases, then you can go on and you can purify it with some sort of like protein chromatography, where you have little beads that are going to separate the proteins based on various properties. So all in all, it's a lot of work, but hopefully it pays off and you get really cool protein that you can then work with. And so just an overview of what we've been talking about. So with this system, we start with the subcloning, we stick it into this donor plasmid. Then we make the backmid, so with the shuttle vector that can go between the bacteria and the insect cells, and have the instructions for making the insect cell virus, but that we can grow in bacteria. And so we do this using with these special bacterial cells that have a helper plasmid that makes the transposase, and so like these DH10 cells. Or there's like different cell types, there's different ways. Some of these use other methods for getting your donor into your acceptor and stuff. So look up the methods for your particular system. Speaking of which, this is and vitrogen has a nice guide, as well as this, the paper for the system that I'm using. But so basically you make this backmid, and now you need to transfect it. So you stick it into the insect cells with giving them some help. So you have these plated cells, you use this transfection agent to sneak that backmid inside. Those backmids will start making virus. This virus and secreting the virus. The virus is gonna have the coat now. So you don't need to use a transfection to get it into more cells. So you filter this out. You have that's your V0. You use that V0 virus to infect more insect cells, and then they start making the virus. Now you have more virus. So this is your V1. You collect that virus, and then you can drench more cells onto it to get lots of protein made. Then you collect those cells, you lyse those cells, you centrifuge those cells, ultracentrifuge them, collect the part that you want, and purify out your protein. And so that is the basis of the Baculovirus Expression Vector System. And happy expressing.